name is Ying. I'm from University of Coimbra, Portugal. Today I'm going to talk the different area. My topic is about Rhizobacterium Bacillus species SC2B and its role in phytoextraction. As we know, mining activity and pathogenesis of ultramafic rocks have caused high metal contamination in many countries. As an emerging technology, phytoremediation offers great potential. Phytoremediation offers great potential to remediate polluted environments. The definition of phytoremediation is the use of higher plants to remove contaminants from the polluted soil. It includes phytoextraction, degradation, phytostabilization, and phytovolatilization. Today, I'm going to focus on phytoextraction. However, it's a long-term process which depends on the plant biomass and the plant's tolerance to metal toxicity and the metal accumulation capacity. Recently, the benefits of combining plant growth promoting Rhizobacteria (PGPR) with plants for enhanced phytoextraction has been proposed. When we're talking about the microbe-assisted phytoremediation, we have to know what exactly happened between the plants and the soil microbes. In general, the plant root exudates offers the carbon sources for the soil microbes. In return, the soil microbes provide the fixed nitrogen and some other plant growth promoting substances, such as indole acetic acid IAA, acropropane carboxylic acid deaminase, ACC deaminase, and solubilized phosphorus. As effective bioinoculants for the microbe-assisted phytoremediation, the PGPR must be able to promote plant growth. The plant growth promotion mechanism include the production of plant growth promoting substances and the increasement of mineral nutrient bioavailability in soil and biocontrol. Namely protection of plants from the phytopassengers. In addition to the plant growth promoting mechanism, some of PGPR can also increase the phytoextraction efficiency by mobilizing the metal bioavailability in soil through through release of chelating reagent, acidification, and redox chains. The proposed mechanism involved in microbe-assisted phytoremediation, including the mechanism of plant growth promotion, detoxification, biocontrol, and high metal tolerance, as well as the mechanism affecting the metal bioavailability in soil and the metal translocation in plants. The success of phytoextraction depends on the biomass of the remediating plants, but also on the uptake potential of the plants. As I discussed before, the PGPR can stimulate plant growth by producing phytohormone and solubilizing insoluble mineral nutrients and suppressing phytopassengers. In another hand, it can reduce the phytotoxicity of metal through production of siderophore, antioxidant enzymes, and the bioaccumulation of metals by bacterial cells. In our study, we use sedum plumbing vesicular as our experimental plants. As a newly discovered zinc and cadmium hyperaccumulator. It has been proved by Wu 2008 to be capable of hyperaccumulating cadmium and zinc. Our study area was located in lead zinc mine area in Cheng'an City, Zhejiang Province, southeast of China. The soil from this mine area has been reported to be highly polluted with cadmium, zinc, and lead. The objective of our study was to isolate and characterize metal mobilizing PGPR that could increase phytoextraction of multi-metal contaminated soil. Among 15 metal-resistant strings isolated from the reservoir of the sedum plumbing secular, 8 PGPR was selected based on their ability to utilize ACC as soil nitrogen source. However, string SC2B showed the relatively higher ability to production of IAA, siderophore, and solubilized phosphorus. In the rotor assay, we inoculate these 8 bacterial strings with Brassica push in the Petri dish. The results turned out as C2B showed the maximum increase in the shoot and the root length, and as well as the vigor index among the all 8 bacterial strings, indicating SC2B has the best plant growth promoting effects potential. By morphological analysis, after 48 hours incubation, the colony diameter of this bacterial string was 1 to 6 millimeter, round and circular. Under transmission electron microscope at 10,000 times magnification, these bacterial strings showed rod-shaped. By the biochemical analysis, we can see this bacteria resist the high concentration of heavy metals, such as cadmium, zinc, and lead, as well as antibiotics, such as ampicillin. In addition, this bacterial string also produces the plant groups promoting substances, such as ACC deaminase, solubilization of phosphate, IAA, and hydrogen cyanide, as well as siderophores. By physiological analysis, this bacterial string was gram-positive, and it can hydrolyze pectin and cellulose. Based on the morphological, physiological, and biochemical analysis, and the 16S rDNA sequence, and the phylogeny, this SC2B was identified as Bacillus species. The similarity was 100%. We also studied the impact of L-tryptophan content and pH on the growth of bacteria and its IA production. The results showed in Figure A, we can see in the absence of L-tryptophan, this bacteria did not produce any IAA. However, when this bacteria was cultured in the medium amended with 2 milligram per milliliter L-tryptophan, it produced a highest amount of IAA. However, with the increase in the L-tryptophan, the negative effects was observed. In Figure B, we can see the pH influence on the IA production and the bacteria growth. Actually, it's very clear this bacteria can grow in the medium, cover a wide range of pH from pH 4 to pH 10. However, at pH 6, this bacteria produce the highest amount of IAA. And a significant correlation was observed between the IA production and the bacteria growth. For the phosphate solubilization ability by Bacillus SC2B, we conduct an experiment to study the ability of this bacteria can solubilize how much phosphorus. Then we can see the maximum solubilized phosphorus was observed after 96 hours incubation with a significant decrease in pH. Then we studied the ability of SC2B grow on the DF salt minimal medium. This medium with very low nutrients. Then it's very clear with ACC, this bacteria can grow very well. Without ACC, it did not grow much, indicating this bacteria can utilize ACC by hydrolyzing ACC to alpha catabolate and ammonia, and utilize ammonia as a soil nitrogen source. For the bioabsorption of metal by Bacillus species SC2B cells in the liquid concentration, in the liquid medium containing different concentration of cadmium, zinc, and lead. The results showed these bacteria cells can absorb large amount of zinc and cadmium and lead. However, it can accumulate more zinc than cadmium than lead. This is probably due to the radius of the metal ion. More precisely, the metal ion, which having a smaller size, is more easily to be absorbed by bacterial cells. Then we also studied the effects of inoculation of this bacteria on soil metal mobilization. The results turned out the inoculation of SC2B, you can see, significantly, not only significantly, huge. It increased the water concentration of water extractable cadmium, zinc, and lead by 16.8, 4.6, and 5.7 fold, respectively. We conducted 2 different types of experiments in order to study of the effects of the inoculation bacteria on the plant growth. First of all, in the phytoagar assay, we use Brassica pool as an experimental plant. After the growth of 45 days in the phytoagar, we can see the inoculation of this bacteria significantly increases the root length, shoot length, and vigor index, as well as the plant dry biomass. In the pot experiment, after we use sedum plumingesicular as an experimental plant. After 75 days growth in the molten metal contaminated soil, the inoculation significantly increased the plant fresh weight, dry weight, and chlorophyll content in the plants. I have to say the soil we use in the pot experiment is non-sterilized soil. Actually, this result is amazing. In the pot experiment, we also measured the metal uptake by plants. Then the results showed the inoculation of SC2B significantly decreased the lead uptake. However, it increased the cadmium and zinc concentration in plants. This decrease in the lead concentration was probably due to the direct dilution of the lead concentration by the increased plant biomass. However, the increased the cadmium and zinc concentration in plants was corresponding to the metal mobilization ability of this bacteria in soil. To summarize, we found the Rhizobacterium can increase the water extractable cadmium, zinc, and lead in soil. And this bacteria can improve the plant performance and the cadmium, zinc uptake of sedum plumbing vesicular. The production of plant growth promoting and metal mobilizing metabolites is a likely mechanism. Finally, this Bacillus species SC2B can be a good candidate for microbe-assisted phytoextraction. This work was financed by FCT. Thanks for your attention.
Questions?
You claim that sedum is very good in absorbing, accumulating zinc and cadmium.
Yes, cadmium.
Let's assume that you go to South China and plant these abandoned mines with sedum.
Okay.
It absorbs a lot of zinc and cadmium. What do you do later on when it's fully saturated?
We have a trick with this plant. This plant not only uptakes the high metal, and it transfers high metal from roots to the shoot part,
Yes.
to the air part. So we can remove the air part and burn them, then refine the metals from the soil, from the plants.
Yeah. So you then, what you do in the end, the zinc and cadmium, you burn it and you have collected easily from ashes.
Yes. Any questions? Because I think what I'm presenting is a little different from others. So it's more related to the environmental problem, high metal pollution, because we have the chemical methods, we have physiological methods, but that kind of remediation technology was really expensive. Sometimes we have the soil cleaning, soil watching. We collect the soil from the very highly contaminated soil, and we take to another place, and we wash that, and then we bring back to the place. But this is really very costly with this method. But it's mainly, phytoremediation, this concept, has been proposed for years. But really, it's very difficult to make it applied in the field. Do you know why? Because if the soil highly contaminated with heavy metals, most of plants cannot grow in this soil. So if the plant died, how could we use the phytoremediation technology? So I want to use this bacteria, microbe, assist plants to do this kind of things. And it's really cost effective, and it can shorten the period of the remediation process.
Thank you very much for your presentation. I would like to ask this particular Rhizobacterium Bacillus sp. SC2B. Yes, this has to be introduced along with the plantation of this particular plant you saw us, or it appears just when you start growing these plants? It appears naturally, or we have to somehow introduce it in the particular environment?
I think we have to introduce, because not this bacteria strain. I have another bacteria strain. When I worked in the Chinese academic Chinese academic sciences, actually, I successfully isolated one very super bacterial strain, and we patented it. And finally, it was very lucky to get authorized. And in this case, so if there is some government or company really want to treat with the high metal polluted soil, and they want to use this technique, they can buy or they can contact me. I can provide this bacteria strain for them. It's quite easy. You only apply this bacteria to the plants. That's quite simple and cheap.
I think that is really interesting studies in your study site. But I would like to propose one question to think about what is the major reason for this area has been contaminated in the soil, especially for the heavy metals contamination in this area. Okay. Oh, okay, so that's the mining operation to get the heavy metal, okay. Okay, so what is the acidity waters has been contaminated by the mining site? Acidity water, yeah, especially when is that coal mining or just heavy metal mining or something like that? Yes. Is it copper mining? Yes. Is it copper mining? Okay, yeah, that makes sense to me. Yeah, thank you very much.
And even some industry, maybe a lot of problem with high metals. For instance, the button.
Okay. So what is the maximum depth of the soil contaminated by those kind of heavy metal contamination? Did you make an investigation of the soil profile or something like that?
Actually, we have the concentration.
Oh, okay.
But I didn't private.
Okay, sure. Yeah, right. Thank you very much.